All right. Hello, and welcome to your remote learning Mendelian and rather non-Mendelian genetics lecture. You get to listen to the lovely sound of my voice. Aren't you so lucky? We're going to go through today and review some really quick things about Mendelian genetics, things we already discussed when we were together in school last, and you've probably practiced now at this point if you did the assignment that I posted on Monday. And we're also going to get into some non-Mendelian types of genetics. All right. So far, when we last saw each other and what we've practiced at this point is under the umbrella of complete dominance. This is a case where traits are either dominant or recessive. And you guys know that in the presence of a dominant trait, a recessive trait is masked or hidden. So for example, in pea plant flowers, you can have the genotype that is homozygous dominant, capital P, and this will express a purple flower as the phenotype. The homozygous recessive genotype will yield a white or express a white phenotype for the flower color. But then if you have a heterozygous phenotype, heterozygous, remember, means one copy of each allele, the dominant or the recessive, you will get a purple flower because in this case, the purple is dominant over the white. Remember that the word allele means different versions of a trait. In this case, the trait is flower color, and there is a purple allele and there is a white allele. We use the capital letter to represent the dominant allele, and we use the lowercase letter to represent the recessive allele. That should all be review for you. So if you had a problem where you were told in pea plants, purple flowers are dominant over white flowers, show the cross between two heterozygous plants. I fill in my parents on the top and the side of the Punnett square. Heterozygous plants, so I need a capital letter and a lowercase letter, or the dominant allele and the recessive allele. And I'm going to fill in my Punnett square, which I know you guys are pros at this point. For my genotype ratios, I have a 25% chance for homozygous dominant. I have a 50% chance for heterozygous, and I have a 25% chance for homozygous recessive, and that's for the genotypes, and that would be a 1 to 2 to 1 ratio. In terms of the phenotype ratios, I have a 75% chance of having purple offspring and a 25% chance to have a white flower offspring. And that would be a 3 to 1 ratio. All right? So that's complete dominance, where a trait is either dominant or recessive. The recessive trait is masked by the dominant one. That's all review from middle school. All right? So we're going to proceed now into some other kinds of genetics that are not simple Mendelian dominant recessive. These are what we call non-Mendelian genetics because we kind of put the whole dominant and recessive thing by the wayside a little bit. You might remember these things from middle school. You might not. It's okay. We're going to start today by talking about incomplete dominance and codominance. In the coming days here, we'll review and learn about multiple alleles and then sex-linked traits. All right? So we'll start with incomplete dominance. Incomplete dominance is, as the name suggests, it is a situation where you have two alleles and neither one is dominant over the other. So we drop the whole dominant and recessive thing and instead we just have two alleles. And let's talk about what that looks like with an example. So in these flowers here, you have red flowers, you have white flowers, and you also have pink flowers. So you have an allele for the red color, you have an allele for the white color. And you'll notice that we don't have the same letter anymore. We're using an R and a W. And you'll also notice that one is not capital and one is not lowercase. In this case, because neither allele is dominant, this is how we represent problems that are using incomplete dominance as the type of inheritance. So in this case now, if you have a red flower, it is represented by two capital R's in the genotype. A white flower is represented by two capital W's in the genotype. And if you have a pink flower, the pink is the heterozygous, or the hybrid, we call it, of the two. It is a blending of these two colors, right? If you mixed red and white paint, you would get pink paint. Same thing here, except we're talking about flowers. If a flower inherits a copy of the white allele and the red allele, you end up with a pink phenotype, or what we call a blending of the two. So because neither the red nor the white is dominant or recessive, you don't get a masking in this case, you get a blending. All right, so let's look at an example of a problem with that type of inheritance. Show the cross between a pink and a white flower. So the pink flower is represented by capital R, capital W, and the white flower represented by capital W, capital W. I already have my Punnett square filled in because I know you guys are pros with how to do that. So if I was filling in and answering the problem for what my genotype and phenotype ratios are, I have a 50% chance of being RW and a 50% chance of being WW, or a 1-to-1 ratio. For my phenotypes, I have a 50% chance of being a pink offspring, and I have a 50% chance of getting white offspring from the cross between these two flowers, or again, a 1-to-1 ratio. So here's another example, very similar, still sticking with these flowers, showing you that you have red and white, and the combination of these two gives you pink. If you were to take your pink offspring from what we call the F1 or the first generation and cross those flowers, so here's my first generation flower, RW, pink, RW, pink. If I were to take, this is again 2 from that first generation, and cross those 2 pink flowers, I have a 25% chance of getting a red flower, 50% chance of getting pink flowers, and a 25% chance of getting white flowers. So what's neat here is that you're crossing 2 organisms that are both pink in color, but they have a chance of having offspring that are not the same color as themselves. So you have a chance of getting red offspring, and you have a chance of getting white offspring. All right, so that's incomplete dominance. Codominance is, again, kind of like what the name suggests. In codominance, both alleles are dominant, so there's no blending this time, not like incomplete dominance. Instead, you get a representation of both alleles in the phenotype, and I think the best way to understand it is to look at an example. So, we're going to talk about chickens. Who doesn't love chicken, right? So you have a dominant black allele, or CB. You have a dominant white allele, or CW. Sorry, my phone just went off. You can see that the way that I'm representing the alleles is a little bit different than what we've seen in the past. So I like to represent codominance and incomplete dominance differently so that I know what I'm doing when I'm going through one of these genetics problems. For codominance, I like to use the big letter to represent the trait that we're talking about. And in this case, we're going to be talking about the colors of these chickens. So I use the letter C. And then the superscript or exponent, you could even call it, letter here is still a capital letter, and it's B to represent the black color. CW, or capital W, to represent white. So in this case, again, no lowercase letters. We are not talking about recessive traits. We're talking about a situation where both alleles are dominant and both alleles are going to be expressed in our offspring or our organism. And you would write that, if you had a blending or the heterozygous version, by writing CB, CW as it's shown here. So let's talk about these chickens. These chickens that we're talking about, or this particular species, comes in 3 different phenotypes. You have the white chicken, the black chicken, or the speckled chicken. And you would apply these letters to the capital C as superscripts or exponents. So let's look at an example of what that would look like. If your chicken was CB, it would have black feathers. If your chicken was CW, you would have white feathers. And if your genotype was CB, it would be black and white speckled feathers, or it would look like this. So in this case, now you're not getting a gray chicken. There's no blending, but you actually have a chicken that has feathers that are white and feathers that are black. So both traits are expressed here. Both alleles are here. You don't have a blending like you did with the flowers. You have a representation of the white allele and the black allele all in one organism. All right, so let's look at an example problem of that. In chickens, the black and white alleles for feather color are codominant. If a homozygous white chicken is crossed with a homozygous black chicken, what would the phenotype ratio for the offspring be? So when you're solving these problems, you always start by establishing what your parent genotypes are. And remember, you can write the parent genotypes on either side of the Punnett square. It doesn't matter. So here's my homozygous white chicken. And here's my homozygous black chicken. And if I go through and fill in my Punnett square, you already know, right? I'm going to get all CB, CW, again, keeping it alphabetical. Generally speaking, you want to keep the letters alphabetical when you're writing out genotypes. And I wrote it out because this is specific to how you would show a codominance trait. And I have all speckled or all of the black and the white chicken. Not a gray chicken, remember, but rather a chicken that has black and gray and white feathers. All right. So this can also happen in cattle. Cattle can be, if you've ever noticed them, if you've ever driven around and looked at them, they can have red hairs all over their body and they look kind of red in appearance. They can also be white, or they can be this third phenotype, which is called roan. And roan cattle actually have red and white hairs all over their body, like this one in the picture here. So this is not a pink cow, right? I didn't take my red and my white cow and get a pink cow. Maybe that's where strawberry milk comes from. Just kidding, right? You actually have instead a cow that has both the red allele represented and the white allele represented, just like on the chickens. And you would write it CR, CW, just like we did with the chickens. Here's another example with Appaloosa horses. So this is this horse here that has this speckled appearance or spotted appearance. They can either be gray or they can be white in this case. And because this particular trait, this color of the hair on them is codominant, when you cross these horses, you don't get a lighter gray, if you will, horse. You get one that actually has both the dark spots and the light spots. So cross a white horse with an Appaloosa horse. So you would have your white horse here, CW, CW, and your Appaloosa horse, which is a spotted version, or the one in the picture here, CG, CW, fill in your Punnett square, and you have a 50% chance of having Appaloosa offspring and 50% chance of having white horses, or white offspring in this case. All right? So those are your examples. Tomorrow we will talk about multiple alleles. I'm going to post a sample set for you guys to do. I'm going to post a video of me doing an example from that problem set of an incomplete dominance and codominance problem so that you know how to get started. Keep up the good work. I do miss you guys. I'm sorry that you have to listen to my voice. I'm sure it's painful, but hopefully we're all back together soon. Thank you.